Hello, everybody. Welcome to the Medicon Valley Alliance Microbiome Network webinar. This is the first webinar that we organize, and we're very happy that you're here. My name is Sophie Labros. You've been in contact with me for registration, but during this webinar, you will only hear my voice, and I'll let the others show themselves and talk. So this is a very special MVA meeting. There will be no welcome drinks, no networking this time, and you are all automatically muted. So the interactivity in this event will be through the online Q&A function. You can use this Q&A function for questions to our speakers. And if you can see the other participants' questions, then you can also comment on them if you'd like. This is, however, a beta version of the Zoom webinar, and maybe not all of you can see the other participants' questions. Anyways, if you have any technical questions, you can use the chat and it will go directly to us as the hosts, and we'll try to answer as good as we can. And just a final comment that in the end, we will broadcast a short poll, and it would be really great to have your answers there. Thank you very much. So we're now ready to start the webinar, and I'm really happy to welcome Petter Hartman. He's the CEO of MVA, for a short introduction. Petter, welcome.
Thank you so much, Sophie, and good morning, everyone. Thank you. Welcome to, also from me, to Medicon Valley Alliance first webinar in the Microbiome Network. It's great to see that so many of you have chosen to be with us today. For us, as Sophie mentioned in the beginning, the networking part is extremely important, but due to the situation with the COVID-19, it's obviously not possible to meet physical, and until the situation stabilizes, we hope that we can continue this way and provide you, all our members, we have actually 300 members, with some high quality webinars instead. And this is just one out of, in total, 5 different networks that we have. I hope that I will be able to share that with you in just a second. You should be able to see this on the screen right now. Let's see if I can. There we go. We have 5 networks. Besides the microbiome network, we also have an oncology network, an R&D network, a medtech network, and an executive club network. And these networks are all open for our members. And if you happen to be a non-member following this webinar, you are more than welcome to contact me or one of my colleagues afterwards, and we will be happy to explain more how you can benefit from being a member of Medicon Valley Alliance. Anyways, besides providing our members with an active networking platform, we also do a lot of other things. And one of the things we do is to facilitate and run collaborative projects within the Medicon Valley cluster. And one of the projects we are working with right now, together with our partners, Copenhagen Capacity and Invest in Skåne, is called the Microbiome Signature Project. And this project is all about positioning Greater Copenhagen and the life science cluster, Medicon Valley, as a leading hub for microbiome research. And my colleague, Sarah Lidé, Strategy and Project Manager at Medicon Valley Alliance, will be happy to give you some introduction and say a few words about the status in the project right now. So Sarah, over to you.
Thank you, Petter, for the very kind introduction. And as Petter mentioned, I would like to take this chance to give everyone an update on the Microbiome Signature Project that was first presented at our previous network meeting. So for the sake of those who are unfamiliar with the Signature Project, as Petter mentioned, the overarching aim of the project is to position the Greater Copenhagen region, which spans Eastern Denmark and Southern Sweden, as a leading and vibrant hub for microbiome research and development. So this is a project co-financed by Interreg, which is an EU body, and is jointly led by Medicon Valley Alliance, Invest in Skåne, and Copenhagen Capacity. What we hope to accomplish during this 3-year period is threefold. Firstly, to attract international talent to the region, both for companies as well as for research. Secondly, to attract international organizations to bring their research activities to the region. And ultimately, to strengthen the position of Medicon Valley as a strong and leading life science cluster in Europe. So since the project started in August last year, one of our key areas of focus has really been to map the region's microbiome-related activities. And the purpose of this mapping exercise is to really highlight and make visible all the great microbiome-related activities and ecosystem actors in the region. We hope that the mapping exercise will not only help stakeholders that are based in the Greater Copenhagen region to be able to navigate the regional ecosystem and establish new connections and partnerships, we also hope to attract the attention of international stakeholders in wanting to be a part of the vibrant ecosystem that we have here in the region. So we've gone about the mapping exercise in 3 ways. We've had a series of one-to-one interviews with about 40+ different regional stakeholders representing industry, academia, and clinical perspectives. And I know that a number of you whom we interviewed are listening in on this webinar. And once again, thank you for taking the time to meet with us and for sharing your insights, which have been very valuable for the team. In addition to this top-down approach, we have also conducted a bottoms-up approach, that is to say, a deep dive desktop-based research and analysis into various databases to get a sense of the kinds of research and clinical trial activity that has been going on in the region. And I will be sharing some of these results shortly. But given that we also like to understand how the Greater Copenhagen region is positioned from a microbiome perspective, we've also benchmarked it against some of the top microbiome hubs that were mentioned in the course of our interviews. With this in mind, I'd like to take the chance to share with you some of the key highlights that came from our analysis of research publications in the region. So as I mentioned, we wanted as a team to really understand what kind of research collaborations have been going on when it comes to microbiome research, the volume of this research, as well as how it fares in terms of quality. So we did a deep dive into PubMed and looked at publications from 2014 to 2019. And what we found was that since 2014, there have been more than 1,000 research papers relating to the microbiome that involve researchers affiliated with the Greater Copenhagen region. And these 1,000+ research papers have in turn involved collaborations with more than 80 countries worldwide, the top international collaborators being the US, Germany, UK, China, and the Netherlands. When looking across the other microbiome hubs that we have used as benchmarks, we see that the region generates a significantly high volume of microbiome-related research versus other European hubs, with Boston, of course, being a clear leader here. But not only are the volumes significant, it seems to be that we are generating high quality research as well. So we looked at publications in Nature journals as a proxy for quality, as a first step. And later on, if resources allow, we might extend the analysis to look at citation impact scores as well. But even with this limited proxy for quality, we can see that the region is author to a good number of microbiome-related publications in Nature. So in terms of Nature publications as a percentage of all publications, the region has 2 times the global average or more, with 6.4% of all microbiome-related publications being published in Nature. Boston is again the leader here at 10%, while the next leading European cluster is slightly below 6%. So we can see that the region is in a relatively strong position in this respect. When looking deeper into the Danish and Swedish context, we can see that the Greater Copenhagen region generates slightly less than half of all microbiome-related research in both countries. However, even though we see a lot of great research from both sides of the border, it seems that the level of cross-border collaboration is currently still quite limited, with only 23 out of the 1,000 research publications having involved researchers from both sides of the border. And we understand that this could be due to differences in structural and funding frameworks between the 2 countries, but we do believe that there is potential for increased collaborations across the border. And we hope that our work to this project can help facilitate that shift. To take a broader perspective, we then looked at the regional ecosystem of actors. And you can see here that the region has at least 80+ organizations working actively with the microbiome from different perspectives, with 40+ of them being companies. And I think that this is quite an interesting finding, given that when we looked across the other key European hubs, while they generally have rather strong academic strongholds, one thing that we have in Greater Copenhagen that stands out quite a bit is really the strong presence of industry. So with all these insights in mind, we're now in the midst of summarizing and refining the findings and doing a high-level assessment of the maturity of each segment within the microbiome. Each segment captures various perspectives, such as type of indication, segments along the food pharma value chain, non-human applications such as environmental microbiomics, as well as supporting technologies such as bioinformatics and the like. And the idea here is to identify our specific strongholds in the field from at least an European perspective that we can then highlight and talk about in various international settings. We're also aiming to highlight the region's value proposition, and that is, what are some of the supporting framework conditions that really make it attractive to do microbiome-related research and development in the region? And a lot of the pillars that you see on the right are well known from a life science perspective. And what we want to do is to clarify the specific connection and application to the microbiome field. With that being said, I would like to round off my short presentation by sharing some of the key next steps in our pipeline of activities. Firstly, on 7th of September, Copenhagen Capacity and Invest in Skåne, in collaboration with Medicon Valley Alliance, are organizing a half-day event that is open to all, where companies and researchers will have the opportunity to showcase their company or project with a 10-minute pitch to an international audience of investors and corporate partners. If you are interested to pitch, you are welcome to send a headline and abstract to Niels Andersen at Copenhagen Capacity by 30th of April, and you will find his email on the slide. Next, on 9th of September, as part of the program for Nordic Life Science Days, to be held in Malmö, the Signature Project is hosting a super session called Microbiome, the New Genetics. We will have speakers such as Rob Knight from UCSD, Per Falk, the President and Chief Science Officer at Ferring, as well as Henrik Bjørn Nielsen, the CSO at Clinical Microbiomics, who will be speaking about the current developments within microbiome research. So if you happen to be at NLS Days, you are very welcome to join in that super session. And of course, all MVA members can benefit from a 20% discount at NLS Days. Both these events are found on the MVA's event calendar, so do look them up if you wish to find out more. Finally, the Signature Project will be launching a talent campaign on the 20th of April, where we'll be focused on helping regional organizations recruit international life science and microbiome specialists. We're looking at, for example, researchers and industry specialists within microbiomics, bioinformatics, biostatistics, and regulatory affairs. Signing up for the campaign is free, so if you're interested, don't hesitate to reach out and we'll be more than happy to help. With that, I would like to say a big thank you from me and my team. And if you have any questions or are interested to find out more, you are more than welcome to contact us. Thank you for your time, and back to you, Sophie.
Thank you, Sarah. That was really nice. I also got one question from a participant. If the PubMed search only included the names of the universities, what about publications that come out of R&D departments from companies in the hubs?
That's a really good question. Our current mapping has only covered universities. So as a potential extension of that exercise, we'll be happy to also include the R&D activities in companies and other types of organizations. But as a first step, we wanted to look at the academic environment and how the research is being spun up from that environment.
All right. That sounds very good, Sarah. Thank you very much. Then it is our pleasure to go to the next speaker. Our keynote speaker for today will be Reinhilde Schoonjans, and she is Senior Officer at the Scientific Committee and the Emerging Risks Unit at EFSA. And EFSA is the European Food Safety Authority. She will give us an overview on the regulatory perspectives on risk assessments and microbiomes. Reinhilde, we're really happy to have you here. The floor is yours.
Thank you very much, Sophie. And let me start thanking the organization for having this opportunity to address some of the questions that we have in EFSA and to outreach to such a numerous and esteemed audience. Thank you very much. I will share my screen. So I present on behalf of the EFSA, which is the European Food Safety Authority. And the outline of my talk is first to address a little bit what is EFSA about, who are we, what do we do, and then to zoom into some current activities that are going on with respect to microorganisms more in general as a status quo of where we are. Further, I will zoom into some future activities that are more focused on the microbiome and the questions that we are having on the table, and to provide you with some useful links to reflect how, what are we looking for and who could possibly contribute to and increase the knowledge in this area from a risk assessment perspective. EFSA is located in the north of Italy as being one of the EU agencies that have been decentralized throughout the Union. The yellow dots are also other agencies, but we consider the ones here, ECHA, ECDC and EMA, to be our sister agencies because they are involved in risk assessment and safety issues for the European consumers. We are just located in the heart of Parma, the north of Italy. We are in the middle of the Corona situation. So again, thanks for setting up this virtual network so that at least our activities can continue and everything is working so nicely. Thank you very much for that too. We are 450 staff members, so it's not such a big organization per se on the payroll. Let's say half of that staff is a scientific staff. For instance, like myself, I'm a molecular biologist, but we also have veterinarians, food chemists, we have data officers, we have all sorts of disciplines needed to cover the remit of EFSA. Together, we also work with external experts that are coming from EU institutions, both academics, as well as research institutions or member states institutions. And per topic, we group these so-called working groups or groups of experts to work with us on a certain output that is requested from us in terms of scientific advice. We only give scientific advice in our opinions. We do not make any regulatory decisions. To do that, we meet frequently, either physically in Parma or elsewhere in Europe or online, and we produce about 5,000 outputs. We were established in 2002 after a number of food scares in order to deliver independent advice, to disconnect from social, economical factors that also lead to certain decisions taken at EU level. Our role is to just focus on scientific evidence and facts and create our opinions on that basis. As said, we work with external experts and they populate the different areas EFSA works on. This slide shows you what is the remit of EFSA. It goes basically from farm till fork and everything that has to do with the food chain, in which we intend to include, or with the term we include also feed for animals, as well as animal health and welfare, of course biological hazard, but also chemical contaminants that can come into our food, of course food additives. food packaging, nutrition as a whole, as well as novel foods and allergenicity, as well as plant health and plant protection products. So here is the agricultural component coming in as well. What are now the activities that we have that are related to microorganisms or microbiomes? On this slide, you find a few examples that we have been listing to make a status quo in the whole of our organizations. Where are we with this topic and where do we want to go next? What do we need? So as an example, in the GMO area, we have, of course, work on genetically modified microorganisms. I use a lot of acronyms, sorry for that. You can always ask, of course, explanation. Or genetically modified plants. We now also work on synthetic biology, both for microorganisms and plants. And we had a colloquium, which is a symposium, on how can omics be used in the risk assessments that we are doing. And from there, clearly, omics is very useful to study metapopulations of microorganisms. Let's call them microbiomes. For plant protection products, we have received in EFSA already active dossiers from applicants that seek market authorization for plant protection products that are living organisms. We have a virus, we have a fungus, and we have a bacterium in the assessment pipeline. The bio has a look at the QPS, Qualified Presumption of Safety, to determine whether a given microorganisms for use in the food chain has a safe history of use, and if it then can bypass, let's say, a further risk assessment. Of course, the biohazard panel also have a very important mandate to monitor antimicrobial resistance, or more in general, the properties for that. The NDA studies novel food, as I said. Then we have the health claims also on under NDA, more particularly for probiotics. And then further panels deal with enzymes produced from microorganisms. And the FAF panel, they also have looked at emulsifiers and artificial sweeteners because the literature is booming with respect to their potential influence on the microbiome. So they are doing literature review to assess which is the knowledge there that can link the use of emulsifiers or artificial sweeteners on the status of our microbiome, and then further on the health status of the host or the consumer. Also, feed additives are produced either with living organisms or with enzymes. At my level, we also looked at nanoparticles because they can have an antibacterial property. And the rest of the organization is also involved, definitely plant health and animal health and welfare, because the health status of the microbiome is important as a proxy for their resilience. So what do we do? What do we want to do next? We have acknowledged that microbiomes are a topic of very expanded type of research. Publications are booming. They are linked to certain diseases, and we wonder, how will this integrate in risk assessment? Do we have to integrate it in risk assessment? So we want to have an EFSA-wide capacity building for this topic, starting from gut and soil. We have already mapped that many organism groups are under the wings of EFSA for assessing their health status. Not only humans as consumers, but also the animals that will be fed with feed additives, for instance, and the plants or the wildlife animals that live in the environment. Because EFSA, as being a food safety authority, also deals with environmental risk assessment. So to start thinking about this topic, we organized a workshop with EFSA Scientific Committee. which is a committee of experts composed of the chairs of all the panels that I showed you before. And together we try to harmonize approaches in EFSA, make sure that we are aligned, and to determine what would be the priorities for EFSA in risk assessment and what would be feasible. So I'm going to talk a bit of that. And then next we proceeded in the writing of a thematic grant specification. This will be a further topic of some of my slides. Of importance is there that we seek consortia that can address some of the questions on the table. With that, we seek, so with this thematic grant in the form of a project, we seek the state-of-the-art on methodologies available to study microbiomes, to study causality for their impact on the host, but also causality of the impact of the modulators that EFSA assesses, the chemicals or the biological hazards that we assess. How do they impact on the microbiome? And we want, under those projects, to receive a roadmap of where can risk assessment advance in the future. With all that capacity building exercise and the knowledge generated in it over the next years, we also hope to influence the EU research agenda, because we spotted until today nothing is really focused on risk assessment in particular, not yet. So the risk assessment questions that came out of that workshop are, for instance, how do we interpret the toxicological studies that we are currently having, of which the results are part of our risk assessment product? What are the metabolic functions of gut microbiomes of the test animals, and how do they compare to those of humans or those of domestic animals? So how can we extrapolate from these toxicological studies with respect to microbiomes? Another example is, how can computational models help us to start assessing impacts that may be caused directly through metabolism, for instance, or indirectly through compositional change of the microbiome? Further, in our thematic discussion, we talked about how can studies be provided to map the variation of the background and the impact of the modulators, the hazards that we are assessing. What can we learn from the work done in ruminants for feed additives, for instance? So I've been in touch with my feed additive colleagues, and they confirm that there is research going on, more in terms of efficacy of the feed additive, of course, but it's acknowledged that a ruminant is a very good organism to study, because their lives depend very much on the fermentations that they need to ensure, and that cannot be harmed. Any impact of a hazard would have a serious impact on the performance of the animal. When does an effect become adverse? So here we thought about, we need to focus on functionality of the microbiome, and there is definitely a need for more causality research. What is now, what is now the real impact, but also how do you establish that the impact is linked to the use of a certain modulator? How, therefore, do you establish criteria before modulation, and when does modulation of a microbiome becomes an adverse effect? And one idea was to maybe map, make a database of what is seen in tox studies and try to make these links. Of course, on my slide here, I want to underline these are ideas that came out of this workshop, not crystallized yet in concrete action points. Another idea was, is the disease now the cause or an effect? To unravel that, again, one needs to look at causality. And ideas are, maybe if you don't want to start from the disease side, one could start from the metabolic pathways side and try to link it to families or subpopulations that are present in the microbiota. These ideas have been reported now already in the minutes of that plenary meeting that took place on the 19th of February. And so you can read it from this link on EFSA's website. And the knowledge at that stage was used now for the writing of the thematical grant specifications, which I will zoom into. And later on, you can expect also an editorial in the EFSA journal later in 2020. And also at this stage, we can foresee the research that will be needed, but it's pretty much linked to what I've said before, to map microbiomes on their function, to study which part of the gut is most important, are the current test modeling still okay, and what expertise do we need to unravel all of this? So reaching out to the adverse outcome pathway community, the statistics community, people that do omics, and so on. So the next step is that we wanted to enter into a cooperation with the EU member states to work on this topic. The grant is now online on the EFSA website, all for you to read through the tender specifications. The EFSA focal points in the member states are the major contacts here to help boost this cooperation in terms of clarifications on the project, but also for consortium formation. The deadline for applications is 30th of June. The project is for a duration of maximum 2.5 years. There is a co-financing scheme involved. That means that maximum 50% of the budget will come from EFSA. The other half has to come from the consortia, and there is a maximum of 500,000 euros foreseen for each lot. What you see on the right-hand side is a screenshot of the competent organizations that can participate in this call. The call is published on the EFSA website. You have to look for this number. The science requirements is expressed in the first file, but also when you consider applying, all the annexes have to be considered as well to check if you are eligible for applying and how to do it. So you will discover in this thematic grant call that we have two lots. One is focusing on gut microbiomes of humans and domestic animals, but a second lot, equaling another maximum budget of 500,000 euros, is focused on environmental microbiomes. We mean to cover plant microbiomes, those of wildlife, but also free living in the soil. And the main objective is again to build capacity, more particularly to evaluate the impact on microbiomes by various modulators under EFSA's assessment. These are chemicals or biological hazards. Second, we want to evaluate the impact of the microbiomes on the host, and in order to determine whether microbiomes can be included in the risk assessment under EFSA's remit or not. When we ask the project the more specific objectives, we ask for reviewing the state of the art and critically appraising the evidence and the technology or the models, all in vitro, in silico, in vivo, for the following. For characterizing the structure and the function of a microbiome, to establish baselines where possible. So which are the methodologies available for doing so? Which are the methodologies available for studying the impact of exposure on the microbiome and to look for causality? What are the methods to study the impacts of microbiomes on the health of their host? Again, focusing on causality. And we do believe that any of these topics may have a considerable overlap. So we welcome also another view. Instead of using this division, one can make also other modulations where there should be overlap between the fields that I have now just depicted. And again, we expect the contractor to provide us with a roadmap to advance research that is needed to address the risk assessment needs to account for the microbiomes, both in gut and the environment. So how can one participate to this call? This slide outlines a little bit the steps before, the steps that need to be done at the national level. And the focal point here is the person that can help you to check for compliance and where the organizations can ask information from. Then the focal point will help to insert you on the list of the compliant organization, and then the signing of the contract can then further take these steps. We have a special tool for that, but also my colleagues for cooperation can help. So here are the useful links again. Check the grant with what we are asking for, the list of the competent authorization organizations from which you saw an excerpt, and the EFSA national focal points for Denmark and Sweden are mentioned here on screen, and I'm sure they can help address any other questions in practical terms. Of course, they're also available should there be a need to have scientific clarifications. And with this, I want to thank you for your attention. I acknowledge the big cooperation inside of my organization, my team members, as well as the broader group of colleagues inside of EFSA. The pictures show that Parma is quite empty for the moment under the corona situation, but spring is ahead of us. There's migratory birds in the riverbed, and we see the leaves growing each day. So we are looking ahead for more jolly times. Thanks for your attention.
Thank you very much, Reinilde. That was a very comprehensive talk. It is now time for us to introduce the Q&A session. So this is going to be completely online. And you can use the Q&A function that you can see on your screen. It might be possible that you can see all questions. And if you can see them, then you can also comment on them. For example, if you think that a certain question is particularly relevant for you, or if you would like to add a related question to the same topic. So we look forward to getting your questions in the Q&A. I already received one first question, so we can just start with that one, Reinilde. It is from a participant asking if EFSA provides advice on pharmaceutical microbiome development.
No, the pharmaceutical products are under the wings of EMA, which is our sister agency in London, and they have a particular framework of assessing. I know in their framework there is a requirement for assessing antimicrobial resistance properties for any veterinarian or human pharmaceutical product. Yeah, they have a specific guideline for that, but it's focused on the antimicrobial properties.
All right, thank you very much. I have a next question from Sven Holman. He asks if there is a collaboration between EMA and EFSA when it comes to understanding adverse effects, also in relation to a pharmaceutical product.
Yes, the capacity building exercise will surely at one step involve a outreach to sister agencies once some insights have been clarified. We are just starting. The interpretation of what is adversity is and should be a fundamental scientific or should be based on scientific criteria. We had in the past a work together on what is the difference between endocrine activity and endocrine disruption. This having in mind, also for the microbiome, deciding on what becomes adverse, that will not only be EFSA's decision. Of course, we work in the global risk assessment community, and some coherence is of course needed. We will tread with care, and we will arrive there in the course of the capacity building, but not just yet. Thank you.
All right, that is great. I also would like to say to the audience, somebody tried to raise hand. that doesn't really help because we cannot give you the word. So if you have a question, you have to type it in the Q&A session. So then I got the next question from Tobin. It says, I work for a company that performs third-party certification of probiotics. This is more relevant if there is international agreement on standards. Is there someone in EFSA to discuss this topic with?
So to ask questions to EFSA, there is always the open box Ask EFSA. It's accessible from the EFSA website. The question is indeed a little bit technical because I don't know what it means, international agreements. I do have colleagues working on probiotics. They work in the remit of the NDA panel. More particularly, they work on novel foods. Yes, I don't know what I can add. Of course, any question that reaches EFSA will be distributed to the person that can best address the particularities of the question. In terms of probiotics, yes, we have a few, but none of them are living organisms for the moment. So also there, I believe that initial steps are explored, but we don't have the full wave of living probiotic organisms received as an application under the novel food legislation framework. I hope this addresses a bit the question.
All right, thank you. That was clear. Then I have a next question from Tova. Are you focusing only on the gut microbiome, or could you potentially also investigate effects on microbiomes of the brain axis, for example?
Of the
gut-brain axis.
Oh, cool. Okay. Well, the human body has more than one microbiome. We start somewhere and we focus now on the gut let's say, to give some direction of what we think we need most. This is driven by the explosion of literature that focuses a little bit on the gut. So no, unfortunately, the other microbiomes of the human body are not yet in focus, but of course we are aware that they are also very important. Yeah.
Okay, I think that answers very clearly the question. I have a next question from Sven Holman. Do you know if the EMA has guidelines in place for reviewing a marketing authorization for a live biotherapeutics product? And which committee would review it? Or do you think they're not ready yet?
I can't answer, I'm sorry. I have not researched that prior to my talk. It's an interesting question though, I'm sorry.
That's perfectly understandable indeed. EMA is another organization, so that is totally fine. I think for now I don't have any more questions in the Q&A. Let's give it another try for a few seconds just to see if people are typing and getting up with new questions. Do you expect a lot of applications for the grant? Do you have any experience with previous grants in similar topics?
We do have experience in thematic grant calls in 2 occasions. They are mostly very practically oriented. One was to establish a bioinformatics platform in a particular context of foodborne pathogens outbreaks. Again, there was a cooperation needed with ECDC and member states. It was very practical. Offers, I don't know if we had a lot of offers. The quality of the offers is something to carefully think about. There's also freedom for the one that will apply. So the thematic grant objectives and specifications are a guideline, but are not meant to impose, because the expertise, of course, lies outside. How many? I don't think there is so many, because typically what EFSA asks for is very specific and very specifically linked to risk assessment. Now, this link is super important. We have to tap into the knowledge of academia, of research institutions that maybe have nothing to do at this time with risk assessment. But I do hope that bridges are being formed in such consortia, because risk assessment institutions do not have the knowledge that they have. So this cooperation is extremely important. We encourage consortia formation so that parts of the job can be distributed also to different partners, eventually also to subcontractors. So if we have one or two proposals, we would be very happy. If the consortia are well formed to cover all what we ask for and can demonstrate eligibility, of course, and can demonstrate expertise.
Yeah.
But the range of proposals, in my experience, does not exceed 5.
Yeah, that's a very good estimation already.
And when I say 5, apologies, I mean the normal procurement and outsourcing contract. This thematic grant is another rank order. It's much bigger. Therefore, we do not foresee. But the message is cooperation. Yeah.
All right. Reinilde, we also talked about that you would try to make available your slides, or at least the parts of the slides that are totally open to the public.
Yeah.
So for all of you, I will, of course, send you an email with the link to the presentation on our website when that is available. And I think there is no further questions coming up in the poll right now. So I would like to start wrapping up then. It's also good to say thank you to all of our speakers. Reinilde, fantastic, great that you could be available all the way from Parma in Italy. Petter, thank you, and Sarah, thank you. You're both in Sweden, so this is clearly a very international and online call. I think it was a very good first experience. I'm really happy that that we managed to do this in such an organized and coordinated way. Reinilde, thanks for this time. I hope we will stay in touch if there is any questions about the grants or other things on EFSA coming up. And for all of you, we would like to launch a poll. It's a very short one, only 6 questions, asking you about your experience with this event. As it is the first time for us, your answers really matter to us. So please take your time to answer the short questions in this poll. And Nicole, you can launch it now. That would be really great. And then I hope that you, for those of you who were not members of Medicum Valley Alliance, that you got interested in our organization, that you might consider coming to other webinars or meetings in the future. And if you do so, then you're also very welcome to become a member of our organization. And we look forward to seeing you soon again, online or in real. That would be the best, of course. Thank you very much. I'll keep the meeting open for another 5 minutes, just so you can answer the poll, the few questions. But this was it for today. Thank you very much for your participation.